Hello my dear students, myself Minu Srivastav, and today we are going to discuss the last topic of the unit 5, that is blood products collection, processing, storage of whole human blood and dried human plasma and plasma substitutes. Okay, the same topic of the unit that blood products and the plasma substitutes as present in the unit 3 also. So somewhat we have discussed in the unit 3 and the remaining part we are going to discuss in the unit 5. Okay, and the MCQ series is going on. Okay, so for the upgrading or other information, kindly join my Telegram channel, the link present in the description below. I am trying to upload all the lectures in English language as well. Okay, in the Hindi language, all the lectures are available in the Hindi language, mixed language, not completely Hindi, mixed language, but I am trying to upload all the syllabus in the English language also, so that all the students can get benefit, but it takes some time, so kindly give me some time. Okay, so without wasting time, we are going to start this topic. This is the last topic of the unit 5. Now this is the collection. For the collection of the blood, either for the testing, either for the donation, the median cubital vein in the front of the elbow, in the front of elbow, median cubital vein is primary choice. Okay, for this for collection, a rubber tourniquet is applied to the upper arm. Okay, this should not be too tight, okay, and it should not remain for more than 2 minutes, because if it's if we are taking for the testing, the rubber tourniquet is bound with the upper arm, but it should remain for the not more than 2 minutes. Okay, then the venipuncture site is cleaned with the ethanol. Okay, and the sterile disposable needle, which has 19 to 21 gauge. This needle is used for the adults and 23 gauge for the children, and ask the patient to tie his or her fist. Okay, and after this, the required amount is collected. When the patient is asked to open the fist. Okay, after collecting, the sterile cotton gauge is pressed over the puncture site, and the patient is asked to press the gauge over the site till the bleeding stop. Okay, so as the blood is collected, the blood is poured into the or enter into the tube containing appropriate anticoagulant. Here I have written the anticoagulant as A.C. Okay, when the blood is taken into the tube is already contain the anticoagulant agent. The anticoagulants is mixed with the blood gently. Okay, we haven't used the vigorous shaking. Don't use this. We have to do this gently, revolve the tube, or otherwise gently just invert the tube. Okay, otherwise if we are vigorous shaking, the hemolysis occur. Okay, so then blood is sent for the testing. If we are taking for the donation, if we are taking the blood for donation, okay, during the donation, the rubber tourniquet is not fitted, okay, because it will give high pressure on the elbow side. Okay, so during the donation, the rubber tourniquet is not fitted on the elbow side, and the blood which is collected in the medial cubital vein and the storage the 4 to 6 degrees Celsius, and till the 21 days you can store. Okay, the maximum blood which is taken out is around 420 ml one time. Means from the donor we can collect the blood is around 420 ml in one time. Okay, and we put the label here. During the collection, during the blood collection, during the donation, the bag is gently shaken. Slightly we have to shaken it, because clumps will form. Okay, so for this we can store the 4 to 6 degrees Celsius for 3 to 5 days. Okay, this polythene bags also contain the anticoagulating agents, otherwise the coagulants or the blood get coagulate. Okay, then the label in this polythene bag or the poly bag contain, or we stick the label, and that label contain the following informations like which blood group of the donor, A, B, AB, O, like that, and the RH group. Okay, the D antigen presence or absence, the RH blood group is the RH positive and RH negative of the two type. Okay, and the date of withdrawal, on which date he or she donate the blood. Okay, and the storage condition, at what storage conditions you have to maintain up to the complete duration. Okay, and the expiry date also it means if you are collecting the blood on the first of the month and that day it will get expired. Okay, so this is the whole procedure to collect the blood and to protect the blood from the toxic or from the other unwanted reactions. Now come to the whole human blood. This whole human blood part completely some part I have discussed in the unit 3rd, the blood products and the plasma substitute. Okay, so I have discussed somewhat in that topic and somewhat here. Okay, so before watching this video, you will go this unit 3rd, blood products and plasma substitute. It will get you beneficial. Okay, now the whole human blood, in the whole human blood, the 45% part is the cellular part and the 55% is the plasma part, that is the liquid part. The cells contain the RBCs, WBCs and the platelet, and the plasma part contain the water and the protein and some the waste materials, and as well as the electrolytes also present, some electrolytes also present. Okay. Now, the proteins are mainly of the 3 types. First is the albumin, second is the globulin, and third is the clotting factors. The albumin maintain the oncotic pressure and the globulin are of 3 types, alpha, beta and gamma globulin, and the coagulating factors are of different types like the fibrinogen, thrombin, prothrombin. There are the different kinds of the clotting factors are present in the blood. Okay, so if we are taking the blood contain that if we are taking the blood and centrifuge, then the blood, red blood, RBCs get settled down, and here the RBCs get settled down and the plasma part will remain upper side. It's a yellow part, pale yellow part. The plasma part will remain here. But between the RBCs and the plasma part, we get the Buffy coat region. That this region is known as the Buffy coat region. Buffy coat region. Sometimes it is green, but sometimes it is the color is the sometimes it's a green due to the presence of neutrophil. Okay, here the Buffy region presents the WBCs as well as the platelets. Okay, so the Buffy coat region means the RBCs get settled down and the plasma receive as a supernatant. But between the plasma and the RBC part, we get the Buffy coat region, and that is WBCs, that contain the WBCs and the platelet parts. Okay, platelet part. Sometimes that thin region contain or the carry green color, that is due to the neutrophil. Okay, so kindly just take a screenshot or otherwise draw this, or that draw into your copy. Okay, now after this we will start the whole human blood, that blood components and the plasma substitute also. Now come to the now we are going to discuss about the whole blood and the other blood components. Okay, one by one we will discuss. First is the whole blood. Okay, the whole blood is collected from, as we have discussed earlier, that from the arm cubital vein. From the cubital vein we will collect the blood. Okay, and at a one time the 400 ml or around 420 ml blood is collected, in which the blood is collected in a proper bag, okay, and a specialized bag, and that typical bag contain around 49 or around 50 ml anticoagulant. These anticoagulants prevent the coagulation of blood. Okay, this blood stored at the 2 to 6 degrees Celsius in appropriate blood bank refrigerator. This whole blood has the shelf life around the 35 days in the CDPA solution. What is CDPA stands for? CDPA stands for C for citrate, and its citrate act as an anticoagulant. Citrate bind with the calcium ion and behave as an anticoagulant. Now P for the, it's a CPDA solution. Okay, CDPA, not you can say the CDPA solution or CPDA solution. Here the P phosphate represent as a buffer solution, and it help to maintain its pH. Now come to the D for dextrose. Dextrose maintain the red cell membrane. It maintain the red cell membrane it maintain the red cell membrane integrity. Okay. Now A for adenine, and here adenine provide the energy for the all the cellular content in the form of ATP. After collecting the whole blood, the proper labeling should be essential, and that label contain the ABO, RH, date of donation, expiry date, and these information that label should be contained. Okay, now come to the uses. The whole blood use in the acute massive blood loss. Suppose that the patient has the excessive blood loss. In that condition, whole blood is the best substitute. Okay, now come to the next uses. It help to replace the blood component, because the blood because in the developing countries it is hard to separate, or the management to separate the blood components is difficult. Okay, so in the developing countries generally, if any component or any blood component is less, for this or to treat this situation, the whole blood is replaced, or whole blood is given. Okay, now come to the risk. What happen if any component is less, but to substitute the component, if complete your whole blood is given to the patient, then there is a risk of the overload in the patient with the cardiovascular disease. Especially the patient which are suffering from the cardiovascular disease, they are highly suffered from the overloading, means the volume may be increased, maybe iron load will increase. So it's a risk of the whole blood if the whole blood is given. If the blood is collected, it should be processed within the 6 hours. Process means for the separation of the component. Okay, now there are the different blood component like red cell component, platelet component, granulocyte component, and plasma component. In the red cell component, there are so many components or so many forms of the red blood cells, like the packed red cells, like the frozen RBCs, like the washed RBCs, and the platelet components had the platelet concentrate. Here the granulocyte component has the granulocyte concentrate, and the plasma component has the fresh frozen plasma. We can say it is FFP also, and the cryoprecipitate and the dried plasma also. One by one we will discuss all the blood components here. First is the packed red cell. The packed red cell, these are the red blood cells in a packed form or in the concentrated form. What is the concentrated meaning? The concentrated meaning is there is no plasma or there is negligible plasma. Okay, the process start within the 6 to 8 hours and prepare from the blood by removing most of the plasma. Okay, so it is known as the packed, or we can say it's a concentrated form. Okay, now how we will receive the packed red cell? By allowing the sedimentation or by the centrifugation process. If it is done by allowing the whole blood to sediment overnight in the refrigerator at a proper temperature 2 to 6 degrees Celsius, by which the RBCs can get sediment over the night, or we can use the centrifugation process. For this centrifugation, we have to maintain the temperature around 2 to 6 degrees Celsius. The temperature is most important point for collecting the blood component. Okay, now the centrifugation has the RPM around 3800 to the 4000 RPM, and the time is the 5 to 7 minutes. Okay, this is a store around the 4 to 6 degrees Celsius. Here the packed red cell or packed red cell volume used in the users of chronic anemia. It reduces the risk of volume overload. Okay, it used in the patient of severe anemia with congestive heart failure. Okay, the patient which are suffering from the severe anemia with the particular heart disease. So if the patient is given the whole blood, there is a risk, or they will the heart failure risk will increase. Okay, so in this case the packed red cells will will provide a good option, or we can say it's a better option as compared to the whole blood. Okay, now you will take the screenshot. Afterwards, we will start the other components like the frozen RBCs, the washed RBCs, the platelet components, the plasma component, the granulocyte component, and so on. Okay. Now we will discuss the other red blood cells component and other component also. Now we will discuss the other blood components. Here we are presenting the washed red cells. Washed red cells are the packed red cells but in the washed form with the saline solution. In the packed red cell, as we have discussed, the red cells are concentrated by the centrifugation, and through the centrifugation plasma is removed, but still there will be some 15 to 20% plasma is remaining in the packed red cell. So if a patient has the plasma protein allergy, so this condition it's very critical for such kind of the patient. So to solve this problem, the washed RBCs concepts will be there. In the washed RBCs, in the washed red cells, the packed red cells are washed with the normal saline if the normal saline concentration is around 0.9%. Okay, so after washing, the plasma protein remaining 15 to 20% plasma protein, WBCs, platelets, and all the other things which are except the RBCs, they are removed. Okay, so these are the concept of the washed red cells is a very much important for those patients who have the plasma protein allergy. Okay, so generally given to the patient who have the severe reaction to the plasma proteins, because some patient have the severe reactions to the other plasma proteins. Okay, so we have to so such patient we have to give the washed red cells. It's a better option as compared to the packed red cells. Now come to the next use. It reduce the incidence of the febrile urticaria. It reduce the urticaria reactions and the anaphylactic reaction. In the patient which have the IgA deficient individual, so such patient has the anti-IgA antibodies. So in those patients, the washed RBCs are the better option as compared to the packed red cell. Okay, now come to the frozen red cell. Frozen red cells are nothing. The RBCs or the packed RBCs are freezed or present in the freeze form with the help of glycerol. Okay, so it can be stored up to the 10 years. Okay, so the storage, due to the frozen, the storage time is increased. Okay, the glycerol act as a cryoprotective agent here, and RBCs and glycerol RBCs and glycerol preparation is present over the -70 degrees Celsius. But before using, we have to increase the temperature, and it come up to the 37 degrees Celsius. So when we are using this frozen RBCs preparation, we have to thaw at the 37 degrees Celsius. Thaw means it's a melting process, or we can say the temperature is increasing. Okay, so you don't get confused about the thaw, that this terminology. Okay, so we have to increase the temperature up to the 37 degrees Celsius, and after this we will centrifuge. Due to the centrifuge, this RBCs get settled down, and these RBCs which get settled down, they washed with the saline. Okay, now the RBCs are ready to use. Okay, ready for using, and these RBCs preparation can use up to the 24 hours. Okay, means if you are removing the RBCs glycerol preparation and you are thawing at the 37 degrees Celsius, so these RBCs you can use, you have to use within the 24 hours. For what they are using? For rare blood groups. Their main preparation for the rare blood groups means the patient, if a person want that I am having the rare blood group incidence, so for this I want to freeze my RBCs. In those cases, the patient can, or the person can froze their RBCs, his or her RBCs. Using the autologous transfusion. Autologous transfusion means you can use or you can froze your RBCs for your own purposes, for your own future conditions. Okay, so this condition is known as the autologous transfusion. For the massive transfusion. For the massive transfusion means, what is a massive transfusion? Massive transfusion means the critical condition, sometimes the war-like condition, the condition present in the country or in the world. Such, for this condition, we have to provide the blood in a massive condition, in the massive form. So, for those conditions, the massive transfusion is important. For those, the frozen RBCs worked very well. Okay, now come to the platelet. Platelets can be obtained by the differential centrifugation of 1 unit whole blood by the platelet pheresis. What is the platelet pheresis? Before going the platelet pheresis, we have to know, we should know about the apheresis. Apheresis. Apheresis means, it is the term if a donor donate the blood with the help of that median cubital vein. If a donor donate his or her blood, but the patient need only particular blood component like RBCs, WBCs, platelet, only some or one part he or she deserve or needs. So what happen? What the doctor can do in this condition? He will differential separation or differentially centrifuge the particular blood component, and remaining complete blood return back to the donor's body. Okay, in this condition, known as the apheresis. So I am repeating again, the apheresis is the condition in which the donor complete blood is not utilized, only the particular blood component is separated and the other remaining blood is returned back to the donor's body again. Okay, so if we are using the platelet component, it is known as the platelet pheresis. Okay, now the formulation of the platelet is the platelet concentrate. The platelet concentrate is that 1 unit of whole blood is centrifuged at low speed. Okay, we have to centrifuge at a low speed to obtain the platelet rich plasma, PRP, platelet rich plasma. The platelet rich plasma is then centrifuged at high speed. Okay, after low speed, this platelet rich plasma is then again centrifuged at the very high speed. In this condition, the platelet aggregate at the bottom, and the platelet poor plasma means the plasma which is very much poor in the platelet concentration act as a supernatant. Okay, and that remain at the top. Okay, now most of the PPP, what is the PPP? Platelet poor plasma is returned to the primary collection bag. What is the primary collection bag? The primary collection bag in which the blood is collected mainly and after this the blood is centrifuged from the primary collection bag. Okay, then this PPP is returned back to the primary collection bag. What is the dose? And 1 unit of the platelet concentrate will raise the platelet count by 5,000 by 5,000 per microliter. Okay, adult dose is generally 1 unit per 10 kg or 4 to 6 unit. Okay, use in thrombocytopenia means the platelet disorders are treated with the help of platelet concentrate. Okay, in the thrombocytopenia, hereditary platelet functions defect, aplastic anemia, hematological malignancy after chemotherapy, means if the patient undergo the chemotherapy or the radiotherapy, he or she can get the hemolytic malignancies. So in case of that, the platelet concentrate is the important option. Okay, we can store the platelet is a 20 to 24 degrees Celsius with continuous agitation for 3 to 5 days. Okay, if we are taking the low temperature, we can use it for more days. Okay, now you will take as a screenshot, and further we will start the other remaining blood products like plasma, frozen plasma, and that cryoprecipitate and other plasma. Okay, so now we will discuss the remaining blood components. Now come to the next preparation, that is the granulocyte concentration. Granulocyte preparation or granulocyte concentration is rarely used and it is obtained by leukophoresis. Phoresis I have discussed earlier. Okay, the differential centrifugation, differential separation of the proper blood component is known as the phoresis. Okay, if we are separating the leukocyte, it is known as the leukophoresis. Okay, or we can use differential centrifugation. It is used in the severe neutropenia. In the severe neutropenia, if the patient is not responding with the antibiotic therapy, then the granulocyte concentration or concentrate is the important or the better option. Okay, it can be stored at the 24 degrees Celsius for 24 hours. Now come to the plasma component. The plasma component can be separated by plasmapheresis or by the differential centrifugation by whole blood. Okay, now first preparation is the fresh frozen plasma. We can name it as a FFP also. Okay, when we collect the whole blood, we can use or separate the fresh frozen plasma by the help of centrifugation. During the centrifugation, the RPM is 3500 and the time duration is 5 minutes. Up to, we have to maintain the temperature 4 degrees Celsius, and then rapidly the fresh frozen plasma is frozen at the minus 25 degrees Celsius or the low temperature. It must be processed when the blood is collected. Within the 6 hours of the collection, the blood is processed for the fresh frozen plasma or the other component separation. Okay, and up to the minus 25 degrees Celsius, we will store it for 1 year, and for minus 70 degrees Celsius for the 5 year. Okay, whenever we will need this fresh frozen plasma, we have to thaw this up to the 30 or 37 degrees Celsius. Thaw means we have to melt or we have to increase the temperature up to the 30 to 37 degree Celsius. Now, as we have increased the temperature, we have to use within the 2 hours. But if we are decreasing the temperature, if we are increasing the temperature around 37 degrees Celsius, then again we have to store it 2 to 7 degrees Celsius for 2 hours in the blood bank refrigerator. Okay, so it means if we are keeping the temperature 2 to 6 degrees Celsius, we will keep it for 24 hours. And if we are keeping the temperature 37 degrees Celsius, we will use within the 2 hours. The dose is 15 ml per kg of the body weight, and the use is in thrombotic thrombocytopenia purpura and the multiple coagulation factor deficiency, as we have seen in the liver diseases. The advantage that if we are giving the patient to the if we are giving the fresh frozen plasma to the patient receive the different factors, factor 8, factor 9 and other clotting factors. and the albumin, globulin, other plasma proteins which are deficient or which may be deficient in the patient can get these also. Okay, now come to the next plasma preparation, that is the cryoprecipitate. Cryoprecipitate is prepared by slowly thawing of the 1 unit of FFP at 4 to 6 degrees Celsius. Means when we are increasing the temperature from minus 25 degrees Celsius to the 4 to 6 degrees Celsius, slowly we have to increase the temperature. During the slowly increasing, the some precipitate start to form, and after this the centrifugation is performed, and after centrifugation the remaining supernatant is removed, and the settle down or the that sediment is frozen at minus 25 degree Celsius. We have to store it minus 25 degrees Celsius for 1 year and minus 70 degrees Celsius for 5 years. Okay, whenever we have to require or whenever the patient require, then we have to thaw, means we have to increase the temperature about the 30 to 37 degrees Celsius, or after this we will keep it in the blood bank refrigerator and 2 to 6 degrees Celsius, but we have to use within the 6 hours. Okay, otherwise it will degrade. We will use in the cryoprecipitate in the hemophilia A, von Willebrand disease and the fibrinogen deficiency. Now come to the dried plasma. Some books refer the dried plasma as the fresh frozen plasma is same, but some books are writing differently. So according to my thinking, they are the similar preparation but slight difference. Okay, so I have written somewhat here. The dried plasma preparation, it's prepared by freeze drying. Okay, freeze drying, the supernatant fluid is separated, and the plasma is pooled with the proper ratio of the blood group. Okay, the plasma is pooled with the blood grouping and to neutralize the agglutinin. Means if any agglutinin is present, so to neutralize these agglutinin, the agglutinogens are added. Okay, then plasma is stored at the 4 to 6 degrees Celsius, and simultaneously properly sterility testings also we have to perform. Then this dried plasma contain the protein content is around 4 to 4.5%, and mainly it contain the fibrinogen. It is stored below minus 20 degrees Celsius for 1 year and minus 70 degrees Celsius for 5 years. When we are using the dried plasma, we have to reconstitute. Reconstitute means we have to make the volume of the dried plasma with the help of water for injection or NaCl injection, or the solution containing 2.5% dextrose or 0.45% NaCl. Okay, and the equal volume of plasma is added. Okay, after reconstitution, if we are reconstitute the dried plasma, then we have to use immediately. Okay, if we are reconstitute the plasma, we will not keep in the refrigerator, and we have to use immediately. Here we have completed the last topic of the unit 5. I hope you have enjoyed the session, and in the English language we will start from unit 1st onwards. Okay, in the unit 5 we have completed most of the lectures in the English language also. Okay, in the Hindi language all the lectures are there, but in the English language only 5th unit will be there. So I am trying to complete from the unit 1st to unit 4th. I will complete in the English lecture within the short time of span. Okay, stay safe, like, share, please like, share and subscribe. Stay safe, stay healthy. Thank you.